Okay, welcome to Gene Expression. This lab starts with a PowerPoint, and I've attached the PowerPoint for you so you can watch, or you can use it in class. So we're going to start right here, and we're going to say, okay, so we've been talking about genetics. We know that genetics code for your phenotype, but how does that actually happen? How does the DNA that we have code for the phenotype that we see in our bodies? And so that's what this lab is all about. So we got to get from DNA to phenotype, and then you want to explain that phenotype has everything to do with proteins. So let's talk about that. So let's say we take the human heart. The human heart is something that your body has to be able to build. Everything that is you has to be able to be built using the genes. So the human heart has to be built, and there's lots of different features. There's blood vessels, there's muscle, there's fat, there's connective tissue. And so each of those types of tissues are built out of a different kind of protein. We are, after all, made out of almost entirely proteins. And so we're going to look a little deeper. We're going to look at two different types of tissue. So we're going to look at muscle tissue, and we're going to look at blood tissue. So you might ask students, do you think that the proteins inside muscle tissue versus blood tissue are different? Or are they the same? Is everything the same? And obviously, everything is different. So we're going to go a little deeper. And we're going to look inside the red blood cells. So inside the red blood cells, we have a very specialized protein called hemoglobin. And hemoglobin, you might point out, bring them back to what we've learned before in lecture, that there is quaternary structure. We have 4 different subunits that are all balled up into their tertiary structure, working together to make hemoglobin. Okay, and this is myosin. We're going to look inside a muscle cell. This obviously is skeletal muscle and not cardiac muscle, but they're very similar. They work with actin and myosin. And this is just FYI information, but it's really interesting that those little myosin molecules, they're like little hockey pucks, and they work like the way that Velcro works, or like the way those little sticky burrs that stick to your socks from a weed, that's how they work. And the myosin actually, when you contract your muscles, it ratchets, and it grabs onto the actin, and it pulls those fibers together, and that's what causes muscle contraction. They don't need to know that, it's just kind of fun information to share with them. So myosin is one protein, hemoglobin is a different protein. So the question is, do you suppose that the DNA that made hemoglobin, the recipe for hemoglobin, do you suppose it's the same as the recipe for myosin, or is it different? Now, obviously, students should be able to answer that, yeah, it's got to be different. And how so? Why does the sequence have to be different? Well, we have to have different amino acids that are going to fold up in different ways to make the different tertiary and quaternary structures that we see there. So we're going to go a little deeper into the primary structure of the protein. So if we unravel it into its primary structure, both of these, we're going to see beads on a string. These beads are amino acids all in a row that form this particular protein. And we've talked about this in lecture, hopefully in all lectures, that the R groups on the amino acids, some of them are positively charged, some of them are negatively charged, some of them are polar, some of them are non-polar. And so when you get the primary structure and you let it go in the right environment, it will wad up on itself to make these tertiary and quaternary structures. And so obviously the sequences are different. Now this is just the very first little bit of these proteins. These proteins are way longer than this. But you can see, even in the first little bit, each of those amino acids, and that's the one-letter code for amino acid. So you've got methionine and histidine and serine, whereas this one is methionine, serine, and then an alanine. So they're different sequences depending on what the protein looks like. Okay, so how does the body translate the DNA, which we know is just 4 letters, A, T, C, and G, into these finished products? Okay, so breaking the code. So we know there's 20 different amino acids. These are the different amino acids with the different R groups. I don't expect students to know these or memorize them, but these are the 20 different amino acids. We have 4 letters of DNA. We have 20 amino acids. So the question becomes, what's the size of a word? Now, here's your first term you're introducing, a codon. A codon is a word. So in the English language, some words are 3-letter words, 4-letter words, 5-letter words, 10-letter words. We do have a couple 1-letter words, right? Like I, A, okay? But that's what we're asking. In DNA, all the words are the same size. How many letters make up a word? And here's our hypotheses. Now, students probably have had this in high school or somewhere, and they know that it's 3, but if students start shouting out 3, I want you to ask them, why is it 3? How do you know it's 3? Why couldn't it be 2? Or 1? Okay, and give them a second to talk about it so they can come up with a reason why. And the reason why is that if you have one, that would mean that A codes for an amino acid, T codes for an amino acid, C codes for an amino acid, G codes for an amino acid. So 2 to the 1. That's not enough. We have 20 of them. So let's try 2. We had 2-letter combinations. So AA is 1, AT, AC, AG, TA, TC, TG. Those are the, so you can go 2 to the 4th, and you can get 16 different combinations. And 16 different combinations is almost enough, but it's not quite. So then you go to 3, and with 3 letters, you can get 64 different combinations. So that's enough to code for our 20 amino acids. But it's way too many. There's 64 different 3-letter words we can make with DNA, and we only have 20 amino acids to code for. So we have some redundancy. So like there's multiple 3-letter words that code for the same amino acid, and we call that factor wobble. That there's a little bit of wiggle room in the number in the words and what they code for. It's usually in that 3rd position, and I'll show you that on the table. All right, so this next experiment, they did this experiment to try to confirm this because they had the hypothesis, well, it's probably 3 letters because that would give us enough combinations to make 20 amino acids. And so scientists set up this experiment where they took a repeating sequence, a 3-letter sequence of DNA or RNA, and they translated it into amino acids, and then they randomly mutated it and added 1, 2, or 3 uracils, which is another base that we'll learn about today with RNA. And what they found is that they got the right sequence of amino acids, we got the right amino acid, and then the wrong one all the way down, and the wrong one all the way down. But on this one, they got the right one again. So does that confirm that it's a 3-letter code? Why or why not? Ask the students, let them discuss. Turns out, yes, that confirms it. You have 3 that will give you a wrong one, and then you get the right ones again. Okay? So, given that information in that experiment, then this scientist named Nuremberg went through all the different iterations of 3-letter repeating sequences that you could do and figured out what every 3-letter code amino acid it coded for. Okay? And so this one, if you do a long strand of U's, you get phenylalanine over and over again. So obviously U is phenylalanine. And he went through that, had a lot of time on his hands, did all of those, came up with a genetic code. And here it is. So this is the universal genetic code. We call it universal because all organisms use it. And this is how you read it. So we've got the first letter of the codon is on the left-hand side, the 2nd letter of the codon is across the top, and the 3rd letter of the codon is on the right. So if I gave you ACG, you would go to A, then you would go to C, then you would go to G. Here it is, ACG codes for 3. Okay? There's one missing right here. This should be asparagine. No, aspartic acid. No, asparagine. No, that's actually wrong. I think it's glutamic acid. Anyway, there's one missing there. I think it's on the next one. Okay, so they should be able to read this code. Now, this is where you can point out wobble. So like, look, that GUU, GUC, GUA, GUG, they all code for valine. So there's some wobble in the code. Okay, and there's wobble here, there's wobble here, wobble here. So you just point that out, that there's way too many 3-letter codes for the 20 amino acids, so we have some redundancy. Okay? The other thing you might point out is the special ones that are in green and in orange. So this one, AUG, it codes for methionine, but it just so happens that all protein sequences start with AUG. So it is the start, but it still codes for an amino acid. And then the other 3, UAA, UAG, and UGA, they don't code for anything. So they're a stop, and we'll get to why in a minute, or how that works. Okay, so there's the code, and in our activity today, we will be using beads to represent our amino acids, and you will have bead boxes. Okay, and I'm not sure if there's 8 of them. We need to make sure we have 8 of them, and we need to also make sure that the beads are all filled up. So the lab tech needs to make sure that we get them all replenished. Anyway, so this is, it matches, so you're going to leave this up on the projector, so when they go to translate, they know which amino acid is which on their beads. Okay, it might be nice actually if we just label the lid, but then they open it. I don't know. It's up there. All right, so that concludes the PowerPoint. I'm going to pause this for a minute and then we'll continue.